still at 0. We're 1. Hi, I am Miss Sloan, and this is a review for my AP Biology students, Unit 7. Everybody have the code that needs the code? Okay. There we go. Unit 7, huge, on natural selection. On the chapters that we did, remember you have chapter reviews. So this, we've been on this unit for over a month. I cannot possibly review the whole thing within this time period. You understand that, right? So go to the chapter reviews for the details. What I see myself doing today is if you have some questions, I will answer them, but also just kind of remind you of everything you need to be aware of. I know we just finished chapter 19 yesterday, so today I made a review for you for just chapter 19, and I posted it in Google Classroom. Did you see it? Okay. And I want to remind you that you need to be able to do both population genetics, the Hardy-Weinberg, right? And I'll remind you of those equations. And you did well there, so hopefully you will do well on the exam as well. And the other thing that you have to be able to do is you have to be able to design a cladogram or a phylogenetic tree. Now, keep in mind, we still have another class period, right? We have tomorrow. We don't have a quiz or anything tomorrow. And Corbin, remind me at the end, because he told me the link isn't working for the PDF. I don't know why it's not linking. If you literally go to the College Board website, they have PDFs now of the lab, so you can go there if for some reason I forget, but I'll make sure that link to the lab works for the BLAST lab. And you just want to get, be ready to go with that tomorrow, and then you'll do your POGL in class. So everybody got the big ticket items? Okay. All right. So first thing I want to do is remind you to go and look, okay? Let's see if I can move myself. Go and look through all the unit expectations for this entire unit. So go to the master hyperdoc and see that you can do all of those different things. Read through it. Look at these big ticket items right here. You could draw on this. Let's just talk through the items we see here, right? So the first thing is introduction to natural selection and natural selection. Who do we think of right away when we think of natural selection? We think of Darwin. And then we can think of, like, remember how we took the time to look at what came before Darwin and fixity of species and those kinds of things. And we looked at some of the evidence that Darwin used. So we have an idea about that. Then artificial selection, what's that about? Just man's involvement, right? Directing. We see that in crops. We see it in dogs. We see it in horses and any animals that we have. That's artificial selection. Sometimes it's done on accident, like what we're doing with antibiotics, and we're killing off all the weak bacteria, and who's getting left behind? Strong bacteria that we don't have antibiotics to. Population genetics, 7.4 and 7.5 are totally intermixed, right? Because population genetics just says you are studying the allele frequencies, right? Studying the allele frequencies. And Hardy-Weinberg is that if you don't want evolution to occur, that it's in equilibrium. Hi, sweetheart. No, sorry. Okay. In Hardy-Weinberg equilibrium, that if you don't want evolution to occur, if you want the allele frequencies to stay in equilibrium, stay static and not change, then remember we talked about the 5 conditions. And if you violate those conditions, and that was the 5 fingers of evolution, remember that? That'll cause those allele frequencies to change. Okay, then next we have evidence of evolution, and common ancestry kind of goes hand in hand with that one, right? When we talked about the different kinds of evidence, what are some things we looked at? Fossils, good. What else? Pardon? Oh, yeah, that's a type of fossilization. So one big ticket item is fossils, and then there are all different kinds of fossils. What's another piece of evidence? Pardon? Biochemistry, so you're looking at the molecular evidence and looking at the similarities. Perfect. What else? Biogeographical, because it makes sense. Remember when we talked about the continents together versus apart? What else did we look at? Developmental. So we looked at how an organism develops during the time that after fertilization, but we also looked at the genes molecularly and how similar those were, right? Like the Hox genes. And one more. Anatomical. And remember, anatomical had 3 subtopics to it. Can you remember what the 3 subtopics were? Homologous structures, not analogous, because that's just similar function, right? So homologous structures, what else? Vestigial structures, which point to our ancestral past, right? Where we came from, right? And what's the third one? Developmental. Okay. So that's evidence for evolution, and that points to a common ancestry. When we look at continuing evolution, is it still occurring? Yeah, we can see evolution. Now, when we see it and you're still the same species, that's called microevolution. But if you change to a totally different species, that's called. And then we had trouble with the species definition, right? Because it required a population to be able to what? Interbreed, which is a problem if you don't breed, right? Or if you are a fossil, we can't tell, right? So we talked about the problem with the species definition. Phylogeny is looking at the evolutionary history, right? And we can use things like cladograms, right? Cladograms, and then all a phylogenetic tree is they've added on what? Time to it. Good. We talked about speciation. Remember the types of speciation that we talked about? What was the most common form of speciation? Allopatric speciation, where you start off with a barrier, right? And then you start out with different initial gene frequencies, different selection pressures, different mutations. So even when you come back together, if you don't interbreed, then you know speciation has occurred. What's another form of speciation? Oh, sympatric, so you're side by side. So it could be little behavioral things or microhabitats, or it could be your what? Chromosomes, okay? Let's remember those 2 chromosome words. Talk about them with your bio buddy. See if you can come up with them. Did you get autopolyploidy and allopolyploidy? Allo, I think 2 L's, and I think 2 different species, right? And a hybridization occurs, but it would not normally happen because there are reproductive isolating mechanisms. Oh, reproductive isolating mechanisms. Let's talk about what would keep a species separate, right? What's the very first one? Starts with an H. Habitats. We don't even live near each other, and we can go bigger than that, right? Ecological, we talked about that. So we live in 2 different habitats. What else? What's the next one in? Temporal. So we want to reproduce at different times or different seasons. Okay. So habitat, we're apart in our habitat. We're living right here, but we do things a little bit different. And then temporal is timing. What's the next one? Behavioral. We don't know each other's dances. What's the next one? Structural or mechanical, right? Means our parts don't fit together. And then what's the next one? Yeah, gametic isolation. Our gametes, like maybe one doesn't have a receptor. Those are all what kind of isolating mechanisms? Prezygotic. What are the other ones called? So we could have hybrid, or the hybrid is very weak, and so it doesn't develop into maturity, or it is sterile. Or maybe the hybrid, the F1 isn't sterile, but the F2 is sterile. See, remember all that. So you have autopolyploidy and allopolyploidy. Allopolyploidy, remember where we were? 2 different species hybridizing. Normally they couldn't do that because the chromosomes wouldn't line up, right? But if they undergo a mitotic event and they double up, then all of a sudden you have diploidy. Yeah? What was that was allopolyploidy. Then we have autopolyploidy, where it's one species and they don't do meiosis right, and so their gametes, if there's their gametes form, and if they have a 2N gamete and a 2N gamete, they get together, they are a 4N, and so they're probably not going to back cross either. Okay, good. Speciation extinction. When we looked at extinctions, right, we had how many major extinctions? 5. 3 of them were in and the last one of the Paleozoic was called the Great Dying. It wiped off most life on. But whenever we had these extinctions, it made what? Room for other species to get in there, right? And so at the end of the Paleozoic era, what did we have an explosion of? And reptiles, right? Then how many extinctions were there in the Mesozoic era? 2. And at the end of the Mesozoic era, that extinction gave room for, the mammals. When did we have a whole bunch of life start coming about? We call it the Cambrian explosion, which is the first period of the, see, there you go. All right. Variations in populations. Remember how we talked about maintaining variations within those populations? Remember we talked about the heterozygote, how it can always mask, right? Even in the Hardy-Weinberg equilibrium, when you did the fish lab, the koi pond, remember you couldn't eliminate an allele. You could eliminate a dominant allele, right? If it's a bad trait, you could eliminate it altogether. But can you eliminate a recessive? I tried to cut my finger off yesterday, just for fun. I thought it would be good to slice through it with a knife, but thank goodness my fingernail stopped it from going all the way through. Yeah, it was super fun. Blood everywhere. Okay, next, origin of life on Earth. We know that one, right? The early Earth, what kind of environment was it? Good, reducing because there was no free. Good, but then how long ago did the Earth come about? Billion years ago, and there were a whole lot of volcanic explosions, and so the gases there, were they organic or inorganic? But we had a lot of energy, and we had UV radiation and geothermal, and then we had isotope radiation, and so it's possible that those inorganic molecules could have become what? Or at the bottom of the oceans you have hydrothermal vents, and that could have catalyzed, right? Especially ammonia. Or that was like an asteroid or a comet seeding the Earth. Does that remember that? Okay, so now we have organic molecules, so we need to go from simple to complex. Let's think about the 4 important organic molecules. Carbohydrates, which are built out of. Okay, let's go lipids next. And we had 2 categories of lipids. What were the 2? Fats and steroids are 4 carbon rings, and fats are glycerol and 3 fatty acid chains. Those fatty acid chains could either be animal or plant, gives you the saturated fats. There you go. Okay, so we said carbohydrates, we said lipids. What's the next one? And those are made out of? Great. And the 4th one? Good. And nucleic acids are built out of? And which one probably came first? Because RNA can act as both a substrate and an enzyme, right? Okay, just checking. All right, so we have our 4 important organic molecules. How could we have built them out of the organic molecules? Same thing as before. Yeah? Geothermal and same energy, right, in order to form those. And then we have to begin to use those to build our first cell. But before we're a cell, we are a protocell. Okay? And you need going around the outside, barrier. So what's going on inside is different than what goes on. Perfect. Okay, and then eventually you have a genetic code, and that would be RNA first, more than likely, right, for the reasons we just said. And when you are self-replicating, then you are a true cell. How many years did it take to get our true cell? About 1 billion years, right? Because the Earth is how old? How old? Billion. You're going to say billion? And then the first cell? Billion years ago, right? Okay, good. And then once we had our first cell, prokaryotic or eukaryotic? Auto or heterotroph? Anaerobic respiration or aerobic? And we said they're a heterotroph, right? Then who would have evolved next after the heterotroph? What kind? Cyclic, right? Because that doesn't split. Right. And then you would have had non-cyclic, where you split water and release. And then you would have had the about 2.3 billion years ago. And then once you had that, then you could we went from prokaryotic, yes, we're going to get there. Prokaryotic to eukaryotic. Good. And now we're a eukaryotic cell. Now we're in the ones of billions of years ago, yes? And because of the oxygen revolution, we now created that what? Protects us from the sun. So we can now go from being unicellular and we can crawl out of the water and get on land. Good. And now we are multicellular, soft-bodied invertebrates. Okay. And what's the oldest era of all? Paleozoic era. Now, get your years. What era are we in now? Cenozoic, and that goes back how far? And we remember that about driving our car. What's the era before that? Mesozoic. It ended, and it started. What did you say? And then before that, 241, and then before that was the Paleozoic era. Remember the Paleozoic era? When did that one start? What are the three things we need to remember? Driving, school's out, dinner time. So as long as you're somewhere in the school's out, 240, 250, you're going to be fine. They're not going to split hairs like that. And then when we have dinner time, what time do we have dinner time? About 5. There we go. So as long as you're in the 540s, then you're going to be fine. Even 550. It depends on what book you're reading. Okay. Paleozoic era. Animal-wise, what did we start with? Invertebrates. Good. By the end of the Paleozoic, what had started to evolve? But to get from invertebrates to reptiles, we had to go through being what? A fish, and then, sorry, I'm trying to flick my tongue out like a frog. No, what's next? Amphibian, and then reptile. Plant-wise, we started as algae, and then we became what? Beginning of gymnosperms. What's a common name for gymnosperm? Christmas trees or Hanukkah bushes. Yes? Okay, in the Mesozoic era, the Christmas trees and Hanukkah bushes came up to all the way early. Good. And animal-wise, we went from early reptiles to early mammals. Good. And then in the Cenozoic era, we can say we went all the way up to humans and then some. They're not the end all, right? And plant-wise, we went from early angiosperms to good. So there you have reviewed the origin of life. Now, what do I have in here? Okay, a lot. So I have the big ticket items for each of the chapters are here. And then I went over, I'm so scared of it seizing. I went over natural selection with you here and how important it is to have variation, right? For natural selection to work on. It's so stressful. Do you remember what we said about fitness? Your ability to pass on your genes and adaptations, yeah? Okay, and then I had, is it seizing or is it just, oh my gosh, no. It's always 7th period. Okay, but let me tell you what I have in there if it stops seizing, which it probably will not. I went through all the big ticket items again, right? And key slides. And if you like have a limited amount of time, it's going to give me a headache. If you have a limited amount of time, you're going to just go through those, right? What does that get you? C, maybe a B, depending on your understanding of it, right? But remember for the exam, and let's think about last exam, that was not all fun experience. That was not rainbows and unicorns for everyone. Just kind of knowing the material as an acquaintance is not good enough, right? You really have to understand it. You want to practice doing Hardy-Weinberg problems. You can Google that, literally Google it. Besides, remember all the reference material I've given you, like a link, and you can go right to my homepage. And there's so many review materials on there. The College Board has review materials on it. Immerse yourself, because today is what day? Thursday? So you have Thursday night, Friday night, Saturday night, Sunday night, Monday night. How many nights do you have to get ready for this? Waiting till Sunday to start this is not your best choice. Now, if you're a Pippinator, if you're Pippin, right, I understand you might have to, but even studying 20 to 30 minutes each day is going to help you get ready for it, right? Okay. And I'm sorry it's seizing, but I will share this with you and you will have it and all the breakdown. But I think we hit all the big ticket items. Is there anything you want to ask me about? Nothing? Okay, yes?
What chapters are on this week?
How could you find that out? On the calendar. Yeah. Everything is listed there. I think it starts at 15. I think it's 15, 16, 17, 18, 19. And you've had 2 labs. And your labs are graded, so I can return those to you tonight if they're not already returned. Did I return them yet? So those are graded, and I'll input those so you have those. And then you have the lab that you're doing tomorrow, which is not difficult. Okay? What else? Anything else? I'm so sorry it's seizing. I don't know why it does that. Anything else you want to ask me? Because I don't want you to come up afterwards and say, hey, can you help me? Just ask it now so in case somebody else needs it. We're good? Okay, I'll see you tomorrow in class. I love you. I love you. I love you.